Right.
So it's wonderful, and it's my pleasure to introduce our speaker today, Dr. Benjamin uh, Gowers, and if I'm not mistaken, butchering it.
That works.
Dr. Gowers actually got his bachelorette in the in the West Coast in Stanford University in biological science. And then he decided that it's too warm there. So I'm just gonna move entirely to the East Coast and spend
Palm trees.
the entire time in East Coast. So there he started getting his medical degrees and PhD. So he's an MD PhD on internal medicine, working on immunology and virology, the fields that you might have heard these days, and it's important: infectious disease, clinician and research scientist, which does the work in the lab and sees the patients. Of course, Ben's work has been instrumental. He has done his postdoc in Elliot Gibbs' lab around 2009, 10, I assume.
Yeah, it was.
Yeah, and then around that time, he worked on basically showing a lot of information regarding how the B cells interact with the EBV, Epstein-Barr virus, and this host pathogen interaction field have been developing. And since then, Ben has been, of course, not only the EBV guy, but recently a lot of work in SARS-CoV-2 infection. He has been a board member of HIV co-infection and other microbiology-related clinical work. He has been recipient of a lot of awards. He has been also an amazing teacher and a graduate program director, associate chair in virology in Harvard Medical School. A lot of the works that he has been done, we have been following for years. He has been a pioneer in having the screening and driving the CRISPR-Cas9 screen in EBV-associated cancer, showing how we could reactivate some of the EBV within the cells, which could be utilized for oncolytic therapy and so on. I don't want to take up so much time, Ben. If you don't mind, share your screen.
Oh yeah, of course. No, thank you so much for that kind introduction, Majid. I have it ready to share, so let me know if this worked okay.
Perfect.
Can you, I just have to make it presenter view,
Yes.
but does that, 1 second, just clicking on that. Does that look good on your screen?
Perfect.
Okay, yeah. It's funny, I'm actually going to have the opportunity for the first time since the pandemic started to give a live seminar in traveling somewhere next week. So it'll be an interesting experience after a year and a half of doing these Zoom ones. I guess wearing a mask. So yeah, thank you, Majid. Thanks so much again for the invitation. I think some of you probably saw Bo Zhao, my colleague, give a talk. Was it, how long ago? I guess that was a long time ago, Majid, right? Like 2 years ago?
It was about, 2 years ago.
I think he traveled there. Yeah, so I guess I'll you gave me the heads up. I should give a lot of introduction and background to the group. So I was going to go through for a number of slides just to give you some history, landscape of Epstein-Barr virus and why you may find it interesting, and then take you from there into what Majid just mentioned in the introduction, a CRISPR screen that we used to understand some of the host virus interactions for what maintains a certain latent state of the virus that I'll tell you about. And then if there's time at the end, so I'll tell you about some recent unpublished work where we're looking at crosstalk between epigenetic and metabolism pathways in the cells and how the two come together. And as we're going, if anyone wants to ask questions and stop me, I'm more than happy to take questions as we go. So please just, if anything is unclear or if you want to ask a question, it'd be great just to bring it up as we go. So Majid, you wanted some background, so that we go all the way back to the beginning. It's kind of a fun history in terms of how do we discover Epstein-Barr virus, because it turns out it was really a tough thing to discover on many levels, one of which it was the first human tumor virus to be discovered. At the time, Tony Epstein, shown here at the bottom of the slide, went after this, it was thought viruses don't cause human cancers. It was known they could cause funny cancers in birds, in other animals. So that was established, but that was thought to be sort of a quirk that didn't happen in people. And so that was one of the huge barriers that he had to overcome. But so was the area that I'll tell you about today in terms of how the virus really restricts itself and becomes very dormant. So he didn't know quite what he was up against, but Tony Epstein at the time, he was beginning his career, had just set up shop in England. And he explains that in those days, you could just open a lab and you didn't know what you were going to work on yet. a little different from today. You would just sort of show up and find something to do. And so he was just beginning, and he saw a flyer that's shown here that he actually saved. I think he had a sense something important was happening. So he actually saved the flyer for the talk that Burkitt came to give that day in London. So Dennis Burkitt, who the eponym Burkitt lymphoma is named after, was a surgeon who practiced. He was originally from Ireland, but practiced in Africa and for some of his career. And when he got there, he started to notice this strange phenomenon he hadn't seen in Europe, where he would see young kids, often boys, with strange tumors in the jaw or face area. And started to realize when he saw it more than once that there was something here that was unusual. And over time, really rigorously studied the distribution of these tumors across Africa. He actually got into, I think he rented a car or somehow got a vehicle and did what he termed a safari, and went across the continent and kind of mapped out where he was seeing incidences of these lymphoma cases. I guess talking to people in the clinics, and sort of realized there was a very interesting geography that it was happening in certain regions in Africa and not in others. And when I teach this to our students, I like to ask them what might this be that's causing this distribution, because that's what really caught Tony Epstein's mind as he sat in the audience this day here in 1961, a long time ago, in the lecture, hearing this talk called "The Commonest Children's Cancer in Tropical Africa, a Hitherto Unrecognized Syndrome." So as Epstein is sitting in the audience, and by the way, I should point out he's young here. He's alive and well. He's 100 years old, still a faculty in Oxford. So he's sitting in the audience and thinks there's got to be an infectious cause of this tumor. Looking at this distribution, and he assumes it's going to be a mosquito-borne illness. And I'm going to go out and find what's this mosquito-borne virus that's causing this distribution. That's what he assumed it would be. He had enough background in virology and even some background in herpes virology that kind of primed him very well. That sort of Pasteur chance favoring the prepared mind was very true in his case. So he set off to go find what that would be, and set up a collaboration with Burkitt for Burkitt to start sending tumor samples from Africa to England for him to start to study in his new lab. He recruited a graduate student who is the other name in Epstein-Barr. This is Yvonne Barr on the lower right side of the slide here. So that was his first graduate student. And unfortunately, this gentleman in the middle gets left off the name of the virus. His name is Bert Achong. He wound up being quite important in discovering the virus. You'll see why in a minute. He's an electron microscopy expert. But because Epstein and Barr learned how to grow and send these cell lines elsewhere that they were culturing, that actually gave the rise to the name of Epstein-Barr virus, and left out Achong in the middle. Epstein has written a lot about how hard it was to find this virus. And one of the big barriers that he had to overcome is that nobody had cultured a human lymphocyte before. It wasn't known if that was going to be possible even, yet EBV spends a lot of its life cycle in B lymphocytes, as I'll tell you about in a few slides. So it took him a long time to learn, how do I actually grow cells from these, what we now call Burkitt tumor samples. And that was a big barrier that he had to overcome. He saved a page from his notebook, and you can see now it's 2 years later here, 1963. You can imagine he's been working all this time for years and not finding the virus. His funding is probably starting to dwindle. He's getting a little bit nervous about his career here. But a couple years in, they get this sample that was really delayed in shipment. I guess there was some foggy weather in England, and maybe that contributed to what happened next fortuitously, where some of the cells from this particular sample had gotten dissociated from the main tumor. They were really cloudy, and he thought that it was actually bacterial contamination. He was just going to throw it out, but thought he would look at it under the microscope before he discarded the sample. And lo and behold, he sees these cells that have now kind of floated away from the tumor, and learned that he could actually grow these in culture. So that was the first time that a B or even a T, any human lymphocyte had been grown. And so he sort of recognized right away it looked a little bit like certain tumors he had seen before in culture. The ascites that he writes here is how you might grow up antibody producing cells in mice if you're going to make a large batch of antibody. So then they go on to work further and take a number of approaches to look for a virus in these samples. And they really had a hard time finding that. And even when they did find it, I'll say on the next slide, he says that this is sort of interesting for, even if you're not going to study virology, I think this could apply to anything that you're doing, that when you're doing something that's so new and a little bit unusual, it can be very hard to publish that. And so he says the paper giving the full account of the unsuccessful as well as ultimately successful procedures had very serious difficulties when submitted for publication. They were reluctant to accept that human lymphoid cells could even be cultured at all. And so he just had a number of barriers. But maybe the biggest barrier was finding the virus. So EBV is a virus that's a little different from, let's say, SARS coronavirus. That might be more what you think about when you think about a virus that goes into a cell and immediately just copies itself. Many copies. When SARS goes into the cell within 8 hours, you have hundreds of virus particles being made and being cranked out of the cell. EBV has a different idea. It's going to live with us for our lifetime. It's not just going to come in like SARS coronavirus for a few days. And so it's got a different strategy where, just like the chickenpox virus, which is another herpes virus, it's going to find a way to get to a reservoir where it can establish latency, shut itself down there, and then pick the time and place to reactivate and finally do what a virus would do and make copies of itself. But Epstein didn't know any of this at the time, so he's setting out to find viruses that replicate. And everything he did couldn't work. He was putting these tumors in the culture and looking for a virus. He was even injecting them into immunodeficient mice or chicken eggs and doing everything he could do to grow a tumor, and everything was failing. And so at the end, he had remembered there were scientists at Rockefeller, I think he'd been there, using electron microscopy to study phage and other early studies of virus particles. And so that was sort of his last ditch effort, was to do that. And lo and behold, in the very first grid of his electron microscope study, he saw in the very first, and then he says he didn't find another one for many hours, but the first grid, he got lucky and found viral particles that he recognized as herpes virus particles in the EM. And so he had an idea now that there was a herpes virus that might be linked to the background of what causes Burkitt lymphoma. It doesn't really explain that funny distribution I was telling you about before. And so the other recipe, just to go back a few slides to this interesting geography in sub-Saharan Africa, is that this is the zone where you find mosquitoes based on the humidity and elevation. And so EBV turns out to be part of the recipe for Burkitt lymphoma when it's found endemically like this in Africa, but the other part is mosquito-borne malaria. And so the two come together in ways that we still don't understand to drive a very high incidence of malaria in these regions in Africa, where Burkitt lymphoma remains the most common childhood tumor to this day. But to go ahead, to give you a little bit more background, EBV fits here in the herpes virus family. There's 8 known human herpes viruses. Still kind of wonder if we're going to discover maybe one more at some point, but as more and more sequencing is done, I guess it's becoming less likely. These may be the 8. And so EBV falls into this branch that we call the gamma herpes viruses, along with Kaposi sarcoma, associated herpes virus. These are the more cancer-causing branch of the herpes virus family, and they're distinct from the alpha branch that includes herpes simplex virus and the chickenpox virus, varicella zoster. We also have the beta branch called that includes cytomegalovirus, and 2 that you probably haven't heard of before, human herpes virus 6 and 7. This one causes roseola. And so interestingly, many of us walk around with at least 4 of these herpes viruses and don't really think about it, but they're in us and we carry them for a lifetime. And so I think this is the last of the big background slides, but I just thought I would take you through. Oh, actually, no, I take that back. There are a few more because Manjit asked me to put in a lot of background. But this is the one that gives you an introduction to the life cycle. So the way EBV, I think, works is that it comes in through the saliva, sometimes it's called the kissing disease. You pick it up. Usually, I think as a kid, I have kids that have gone through daycare, and you see kids putting everything in their mouth and sharing it with other kids. It's like naturally, it's a virus that's acquired very early in life, maybe between families or between kids. And it's actually late, it's really the tail end who evade it as a kid and pick it up in maybe teenage years. They get mononucleosis. which is caused by acute Epstein-Barr virus infection, usually in young adults. And so the virus comes in across the epithelial barrier in the tonsil in the back of the throat area, goes across the epithelium. So this is, where you're going to get a difference from SARS, which would start to replicate here and do its thing in the epithelial cells. Instead, EBV kind of passes right through them and finds the B cell in the tonsil tissue. Our tonsils are areas that are really looking closely at what's coming into the body. What are we breathing or eating in? And so they're taking a close look at that. And so that's where EBV goes to find the B cells that it wants to infect. And here's where things are so different from coronavirus. Instead of making copies of itself, it goes right into these programs that we call latency, where the virus inserts its DNA into the nucleus. That's kind of shown here as this little ring, this orange ring here. And instead of making copies of itself, it starts making these two oncoproteins or antigens that reprogram the B cell. And so EBNAs or Epstein-Barr nuclear antigens. They are transcription factors that either directly bind to DNA in the case of EBNA1, or in the case of these other EBNA proteins, they bind to host transcription factors that lead them to specific spots, either on the virus genome or more commonly on the host cell genome. And so there's a series of these we'll talk a little bit about that are expressed as you take that resting cell and wake it up. The naive B cell is just sitting there waiting for a signal. And it has the neat property that it can just hang out there in the G0 cell cycle state doing very little. But when it gets the activation signal, it can become very activated metabolically and within a few days start to grow very quickly. And so this latency 2B program is probably what the virus is using to wake up the cell and quadruple its size, really start to wake up a lot of metabolic pathways and get it ready for very fast cell growth. Then as these cells travel into specialized areas in tonsil tissue, there's some lymph nodes, and within the lymph nodes, there's specific areas called germinal centers where B and T cells go to really optimize themselves. In the case of B cell, that's where the antibodies get better. It's where mutations are introduced into the antibody to figure out which B cell will ultimately have the best antibody to go after whatever pathogen has infected us. And so EBV has figured out a way to trick the B cell into thinking it's going through that whole germinal center reaction, and it should be growing as it's getting what it thinks are signals from seeing a pathogen, as well as getting help from things like T cells. And so it turns on this full transforming protein called latency 3. So this has also these EBNA proteins, Epstein-Barr nuclear antigen transcription factors, non-coding RNAs. And then it's got these two other proteins called LMP1 and LMP2A. So the full name is latent membrane protein 1 and 2A. They're shown kind of here in purple and red. So they really trick the B cell. LMP1 goes to the cell surface and mimics a receptor called CD40 that normally is one of the main ways that a T cell activates a B cell. So it's telling the B cell, oh yeah, the T cell is telling you to grow right now. And then this red one, LMP2A, is mimicking the antibody on the cell surface that would be seeing an antigen and telling the B cell to get activated because it's seeing the antigen, the thing that it's looking for. And so basically EBV is sending these signals into the B cell to trick it into thinking it's getting all the right activation stimuli in the germinal center and it's time to grow. And so this is the program that if you just infect EBV in culture, you reach this program and it actually immortalizes the B cell because it's making it grow so continuously that they can just grow forever in culture. Thankfully, that doesn't happen to us. Otherwise, all of us would have tumors. And so EBV has worked out a way to expand itself transiently, but then get to where it's going, which is the memory B cell. And so to get there, it starts to turn off some of these oncogenes or oncogenic proteins. And so it goes to this latency 2A program next, where you can see now it's lost some of these EBNA proteins. And then that's thought to happen briefly in this germinal center region. And finally, it gets to where it wants to be, which is the memory B cell. So if you don't have much immunology background, memory cells are what we make. It's what you're making when you get that. It's part of what you're making when you get your COVID vaccines, because you want to have the ability of a pool of cells that are going to remember that vaccine that they saw and be primed to respond very quickly the next time that they're challenged. And so EBV knows that that's a good place to go because these are very long-lived cells. And so if it can establish latency there, that's a great place to hang out for many years and then wait for the time and place to reactivate. So when it gets to the memory cell, it uses this what we call latency 1 program, where it's only expressing a single viral protein called EBNA1. That's what I'm showing here in this red dot. It tethers the viral double-stranded DNA plasmid or genome in the nucleus to our chromosomes, so that if that memory cell divides, it's got a mechanism to spread it to each of the daughter memory cells, so it won't be lost from either one of them. And then it has a neat trick where the memory cell can actually be non-dividing and just be waiting, and it can turn off even that EBNA1 protein. So all it's basically doing is making some non-coding RNAs and sitting in the nucleus as a DNA. It almost feels to me like a retro or lentivirus, where it's just a piece of nucleic acid in the cell nucleus, but completely hidden from the immune system that would be looking for any foreign proteins or anything that it could detect. And so it's a smart strategy for a virus to find a hiding place in the body so that it can affect us for our entire lifetime. Ultimately, it has to be able to do more than this. And so it's thought that when the memory cell gets activated and turns to a plasma cell, which is the type of B cell that makes antibodies to give us our antibody immune responses, that's thought to be the time that EBV knows to wake up. It's smart. It knows that this cell has stopped dividing. It's going to a mucosal surface where it can spread itself to an epithelium. And if this is happening in the tonsils, that means it has a way out to the saliva to get to the next thing and complete the next person and complete the life cycle. So we got pretty interested in the last few years about how does this work when the virus is sitting in this latency 1 state, which happens in memory cells, but also happens in certain tumors. I'll tell you about what keeps it in this state. If you can go to a latency 0 state where it's not making any proteins, or even in this latency 1 state where it's just making 1 protein, how does the virus stay in this hidden state where everything else from the viral genome is turned off, in contrast with this lytic cycle where the virus is making 80 different proteins, or even this latency 3 state where it's making 8 different proteins? So what are the controls that do that? And we were guessing it was going to come down to host epigenetic factors that were modifying viral genome. And we set out to find those that I'll tell you about in a second. But wanted to just say that one reason we're interested in these different epigenetic states is that there's sort of a mirror of them in various human cancers that are associated with Epstein-Barr. So this latency 2B state shows up in certain AIDS, diffuse large B-cell lymphomas. Hodgkin lymphoma, it turns out, has this latency 2A state in the tumor cell. Latency 3 shows up in lymphomas that grow out in immunosuppressed people like AIDS patients or people who've had a transplant and have to get immune suppression after transplant, perturbing the immune surveillance. And so these cells can grow out as tumor cells with the latency 3 program. Turns out that latency 1 is the program in Burkitt cells. And so this is why Epstein has such a hard time finding this virus, because he's looking for evidence of a virus in a cell that's turned off everything but 1 protein, and it's not making many copies of the virus. And so that's why he had so much trouble. And it took him years to find the virus. And I think the way he got a little lucky to find it was that occasionally these latency 1 cells can just reactivate spontaneously. And he happened to bump into a situation where one of these Burkitt cells had switched towards making some infectious virus, and that's what he saw in the electron microscope. To give you a window into how this might look on the viral genome, I think that, as I'll tell you about today, DNA methylation is an important part of what's controlling these different programs on the viral genome. But really not much was known about what are the epigenetic enzymes that had to be coming from the host cell that are controlling these progression of changes on the viral genome. But it was known that when EBV first comes into a B cell, it would use a particular promoter called WP that just drives a couple of these EBNAs when it first comes into the cell. And then as it establishes latency and starts making the cells grow really quickly, it's now turned on a bunch of the EBNA genes. It's now using a promoter called the C promoter. And then from there, it's using methylation as shown in these green sort of balls here to turn off some of the other promoters, including the ones that would drive the lytic cycle, or the original one that was used when the virus came into the cell. And then when it gets to latency 3, another promoter turns on that drives those LMP things I was just talking about, the latent membrane proteins. And it's thought that then as the virus moves towards the latency 2 and ultimately the latency 1 state, you get more and more of this methylation showing up on the viral genome. It turns out, interestingly, that happens on our own cell chromosomes also. And so certain EBV-infected cancers like Burkitt, and especially another cancer I forgot to say in the last slide, there are cancers in the epithelial cell part of the life cycle that are extremely highly methylated on the host DNA as well. Those include nasopharyngeal carcinoma, that's the most common cancer associated with EBV, I think, worldwide, as well as another one. 10% of stomach cancers have Epstein-Barr, probably because we're making it and swallowing it all the time and it winds up in the stomach. And so it's actually present in 1 in 10 stomach cancers. And so those are the ones with the highest level of DNA methylation out of any human tumor. And so it's not clear yet if that's a immune response, sort of an innate immune response to the virus, try to shut it down, or if it's sort of a byproduct of this biology where the virus is using DNA methylation to regulate its own gene expression. So we got interested in the question of what when Burkitt cells have EBV infecting them in this very repressed state, where methylation is shutting down most of the viral genes, all but one of them, what keeps it in that state, and could we push it back to the state where more proteins are expressed off the viral genome so that they become more visible, either to our own immune system, or if it's the immune suppressed host who can't see it, if you could bring in T cells through what's called adoptive immunotherapy approaches, where you can actually raise a T cell in culture and give it to the person. Could we push the virus to a more visible state and then attack it with transferred T cells? And so just to show you again why that would be interesting, in latency 1, the tumor associated with Burkitt lymphoma, there's very low immunogenicity because most of the viral proteins are hidden. But as you move towards latency 3, you actually express more viral proteins and they become more visible to the, or immunogenic to the immune system. So with that background, that brings us to a CRISPR screen that we took on to see could we find what are the key proteins on the host side that repress EBV and keep all but one protein turned off. We use a CRISPR knockout library from the Broad Institute across town. They've made a really nice library in which they have 4 of these called single guide RNA, or sgRNAs, that program the Cas9 protein to make a knockout. So the way we do this mechanistically is we have a Burkitt cell that we can grow in culture. We've stably expressed the Cas9 protein, so to do CRISPR editing in these cells. And then we infect them with this library of single guide RNAs. And so the way this is designed is that each lentivirus that we'll bring in, just one of these single guide RNAs, basically programs Cas9, ideally to cut just to one spot on the human genome. And where it cuts, you'll get a double-strand DNA break, and then the cell will try to repair that very quickly. And usually that is a very error-prone pathway that will functionally knock out the gene that cut was made in. It's called the non-homologous end joining pathway, and very often you get stop codons introduced or frame shifts. And so by making a cut in the gene of interest, you can usually turn off the expression at that gene. There's 4 different guide RNAs targeting every human gene in the library, so that 4 different times we can ask if you get the same effect in your screen. And so altogether, there's something like 80,000 guide RNAs in this library, but you infect the B cells at a very low multiplicity of infection of 0.3, so that on average, a B cell is just infected by 1 lentivirus, it gets 1 guide RNA. And so you hope that just 1 gene is knocked out per cell across your library. And so then we had to take the strategy in this CRISPR screen of how are we going to know if the virus has moved from the latency 1 state to the latency 3 state. And for that, we used a surrogate marker that we could see by flow cytometry or FACS. We knew that in the latency 3 state, the latent membrane proteins are expressed and they turn on cell surface proteins. One of them is called intercellular adhesion molecule, or ICAM1. And we had a nice antibody to that. So we could see if that turns back on, there's a chance that's happening because EBV has switched from latency 1 to 3. And so if we use FACS, we can pull out the cells, we can sort for the cells that have gone from a low state of ICAM expression that the Burkitts normally have to a high state of ICAM expression. We also knew that this other marker, CD10, would go down as we move from latency 1 to 3. So we thought we'll look for that also. But we'll be a little careful with that one because maybe some of the epigenetic mechanisms that we're looking for might be involved in actually silencing CD10. So we might miss it if we just look for that. So we sort of use the sorter to look for different bins. We start out with cells over here in the lower right that are high for CD10 but low for ICAM. And then we're looking for mutations introduced by CRISPR that move the cell from this state maybe to this state where now they're expressing the ICAM1 protein, and maybe they still have CD10 because maybe that couldn't be silenced now. Or ideally, we'd go over to this state where they actually express CD10 and they've lost, they've expressed the ICAM but lost the CD10 as a surrogate for maybe having switched from latency 1 back to latency 3. We weren't sure if this would work, but we thought we would give it a try. And so the way that this also works is that you take the sequences of the sgRNA or single guide RNAs that you have at the input right before you do the FACS sorting. Remember, lentiviruses have brought these into the cell and they integrate into the B cell genome. So that becomes a barcode for what's in the cells. And so you can actually PCR amplify that and do DNA sequencing to figure out what was the abundance of the guides in your library right before the FACS sort. So you know that. And then what was the abundance of the guide RNAs that wound up in the sorted populations. And then you can do statistical analysis to look for enrichment or depletion of guide RNAs. And there's an algorithm called STARS developed at the Broad Institute that looks at whether you got more than one of these 4 guide RNAs coming out in the screen. Ideally, you'd see all 4 of them showing up in the sorted population to tell you that 4 times something happened very consistently. And even if you knocked out the gene in different spots and maybe they have a different off-target profile from one another, you still got the same effect in the screen. And so when we took these sorted cells through DNA sequencing and amplification and that algorithm called STARS, we got a really nice result where there were really 2 standout hits in the screen. And it's nice when this sort of thing happens where the 2 hits called UHRF1 and DNA methyltransferase 1, that'll tell you a lot about, actually talk to each other, they're binding partners. And so the genetics found a nice, basically pathway in which 2 components are very closely interacting with each other. And so that's a good sign that the screen is probably working because it's finding a very nice statistical signal in an area that makes sense. They're epigenetic enzymes that control DNA methylation and they talk to each other. There were other hits that I won't have time to tell you about today from the screen, but we'll just focus on these really strong hits that we got to know through following up this screen. And this is one of the reasons I really like these CRISPR screen approaches, because you take a very large, unbiased approach. You try knocking out genes one by one and just let the genetics tell you where to focus. So going into this, I'd never heard about this UHRF1 before, but it turns out to be a very interesting epigenetic reader and writer protein. So that's just in this next slide here. So UHRF1, I'm sure a lot of you haven't heard of this before, is shown up here in the upper right. So it has a lot of interesting domains. It has a ubiquitin-like domain here, and then it has this tandem tutor domain that looks at the state of histone 3 lysine 9 methylation. So if you have this methylation mark ME2 or ME3 on histone 3 lysine 9, it usually happens in areas of repressed chromatin, maybe at telomeres or maybe in heterochromatin. So UHRF1 is looking with this domain to say, okay, is this a region of DNA that looks like it should be suppressed because it's got a mark on H3K9 that says this is a silenced area of chromatin. It then has this PhD domain that again tells it that it's looking properly at histone 3. And then from a DNA methylation standpoint, it's got this really neat domain called the SRA domain that's looking for DNA hemimethylation. So that happens usually when you've just copied a new strand of DNA shown here. Actually, I should make sure I'm using the right pointer for you guys. So on DNA, the parent strand has the methylation, but you've just copied a daughter strand. And so that hasn't yet gotten the methylation mark. And so this domain is looking for a mismatch. If parent has it, but daughter doesn't have it, then, oh, it must be a place where it's time to copy. And so if it sees this combination of this mark and the mismatch in DNA methylation, then it actually uses this ring domain to add ubiquitin. It's very complicated. It adds ubiquitin onto another histone, and that finally recruits DNA methyltransferase 1, the other big screen hit, to come in and propagate the mark from the parent to the daughter strand. And so this is a mechanism by which when a cell divides, it can remember the epigenetic state of where it began and make sure the daughter cell has that. And so we think that EBV is just taking advantage of this because every day the Burkitt cells are dividing. They're the fastest growing human tumor. I think this would be true as well in memory cells. And so when the cell divides, the cell machinery copies the viral genome. And so when you make new viral episome or plasmids, those have to be reprogrammed to remember what state they were in epigenetically after you've made a new copy of them. And so I think EBV is using this machinery to copy the methylation marks that were there before onto the newly made plasmid EBV genomes. And if you get in the way of this, you'll lose the methylation, and it will then lose the repression and revert to a less repressed state, such as the latency 3 state, where 8 proteins are expressed instead of 1. And so we could see this when we use Western blots. There's a neat tumor sample called Mu2 that began in latency 1 state, but there's a clone of it that had moved to latency 3 in culture. And so you can see on Western blot, you see these EBNA proteins in LMP1 made in the Mu2 but you see how they're not made in the Mu2 that has the latency 1 state. And if we use CRISPR to knock out this UHRF1 protein with either of these 2 different guide RNAs from the library, you see how we lose the expression of the protein. It goes from a high state to very little UHRF1 that's left after we've knocked it out. And when you do that, you start to see that the Mu2 cell now regains the ability to express EBNA2, EBNA3, LMP1, and it can even turn on TRAF1, which is a target gene of LMP1. So you know the LMP1 is actually signaling in this case. Interestingly, it loses the expression of BCL6, which is a major regulator of B cell development. And so we were seeing interesting things happen to the B cells when we did this knockout. We could just quickly, I'll mention, we could use a CRISPR knockout and an add back approach where we take away the UHRF1 and add back either the wild type or versions that have deletions that get rid of any of these different domains in the UHRF1. And when we did that, we could see that they're all important. If we took away the full length and added back a version of UHRF1 that lacked any of these different domains, they couldn't suppress latency 3. So basically all of these were found to be important. And we could be a little careful with time since it's 110, but basically we did an experiment going back to that ICAM1 readout. And if we knocked out UHRF1, we put back the full length, we could suppress the expression of the ICAM1, but if we put back in the deletion mutants that lacked any of those domains, they could no longer suppress ICAM1. So it sort of told us that you need all of these different domains of UHRF1 to keep EBV in this very restricted form of latency. And so from there, in cell culture, we could see something nice happening as well. This is how Burkitt cells normally look. The way that I was showing in Epstein's pictures, you have these cells that grow in suspension in culture. They're just usually single cells, or they grow in very small clumps. So that was true in the Mu2 cell that we used for the screen. But when we knocked out UHRF1, it starts to form these large clusters of cells that look a lot like these lymphoblastoid cells, where EBV has used its latency 3 program to transform and immortalize a B cell. So it was really remarkable to see just this one CRISPR knockout could change the morphology of the cell from what was recognizable as a typical latency 1 morphology to these clusters of cells where adhesion molecules like ICAM1 are being upregulated and causing the cells to stick to one another in culture. And then by immunofluorescence microscopy, we could see when we knocked out UHRF1, we could also see, just like by Western blot, the nice derepression of LMP1 and LMP2A going to the cell surfaces of these cells, as they would in the latency 3 program, but not typically something you would see in a Burkitt cell. So by RNA-seq, we could see something very similar happening with the UHRF1 knockout to what we saw when we just compared the Mu2 to the Mu2 tumor. So again, Mu2 with the original Burkitt in the latency 1 state, a clone moved to latency 3. So they're thought to be isogenic, just differing by the EBV program. And so you see some characteristic changes when you cross compare what happens in the Burkitt cell that has latency 3 versus 1. And when we knocked out UHRF1, we saw a very similar pattern to just comparing the Mu2 to Mu2 tumor. So shown in red are a bunch of interferon stimulated genes that for some reason we've noticed the latency 3 program activates. So they were going up very similarly with the UHRF1 knockout to when you just compare a tumor that's latency 3 versus 1. So sort of supporting the evidence that the knockout moved it from latency 1 to latency 3. And the same thing was happening on the downregulated side. And then we could zoom into the viral genome in these cancer cells and use a technique called CHIP qPCR, or chromatin immunoprecipitation and PCR analysis, to see what's happening at these key promoters that regulate what's actually going to be expressed from the EBV genome, shown at the bottom here. And so in the case of the C promoter, which I have here on the episome, this is the promoter that really drives the latency 3 program. So if you're in a latency 1 cell, you want to silence that promoter. And you can see that there's a lot of this H3K9 silencing chromatin mark that I was telling you is one of the things that UHRF1 looks for. So we see that at a very high level on the C promoter in the Mu2 cell in latency 1. but in black shows you the level of that histone epigenetic mark in the latency 3 cell. So you see it's much lower. And the same for UHRF1. We can see it's actually binding in the C promoter region in the tumor cell with latency 1, but less so in the tumor cell with latency 3. The same thing is basically happening at the LMP promoter that drives the expression of those proteins that are expressed in latency 3. And then getting back to what happens when we knock out UHRF1 using a technique called methylation DNA immunoprecipitation, or MEDIP qPCR. So you're using an antibody against methylated DNA to fish out pieces of DNA that are methylated after you've kind of sheared them by sonication. And then PCR to tell you what's happening at a specific region on the genome. And so in this case, we looked at what was happening at the C promoter. So in latency 1, there's a very high level of methylation that's silencing this latency 3 driving promoter called the C promoter. But if you knock out UHRF1 by either of these two guide RNAs, it just wipes off that DNA methylation mark. It's completely gone from the viral genome. We also got as a screen hit the partner of UHRF1, DNA methyltransferase 1. And again, if we knock that out, not quite as strongly, but we could see a significant loss of methylation on this promoter. So mechanistically, it sort of told us that these are important for maintaining that histone mark on the viral promoter to keep it silenced. If we knock out DNA methyltransferase 1, we could see why it's scored in the screen. This is now showing you kind of how the screen really worked. We were looking for expression of this intracellular adhesion molecule ICAM1 on the cell surface. And if we knock out DNA methyltransferase 1 with either of these two guide RNAs to program Cas9, you can see the expression of the ICAM1 coming up on these orange and blue traces on the cell surface. And by Western blot, we could see, I think I lost my labels, but this was an expression for LMP1 coming back with the DNA methyltransferase knockout. Maybe they got animated. So one thing we really liked was we thought this could tie into the B cell biology and how EBV would know when the right time and place is to make the changes in its latency programs. And so it turns out people have profiled expression patterns across the genome or proteomes as B cells develop. We did this in part, so we actually took primary human B cells from the blood bank, infected them with EBV, and then used a proteomic approach to just look across the whole cell to see what was changing at what time point. And so on the x-axis is time post-EBV infection in cell culture, day 0, 5, 10, 15. On the y-axis is a measure of relative protein abundance across the proteomic experiment, in which we're using mass spectrometry to quantitate proteins across the cell. And so we could see that within 4 days of EBV infection of the B cell, UHRF1 expression goes up very nicely. So does DNA methyltransferase 1. There's another DNA methyltransferase called 3A that also goes up a bit in expression. And this one's interesting because it's one of the things that can actually initiate methylation marks on DNA. So whereas these two just copy marks that are already put there by something else, something has to start this, something has to initiate the process. And it's thought that these are the initiators. DNA methyltransferase 3A and B are thought to be what actually starts the methylation of DNA that these will later copy. And so 3A comes up when EBV infects a cell, but not 3B. When you look at what happens in the tonsils, it's a little different. So UHRF1 does come up really nicely in the germinal center cells where EBV would be just starting to silence the latency 3 program. So does DNA methyltransferase 1. So they're expressed in the right time and place. Now 3A actually isn't. So 3A goes down. So it's there, it's expressed in the resting cell. It actually goes down in the germinal centers where EBV would have latency 3 and start to silence it. But interestingly, 3B goes up right there in the germinal center. So we started to think, oh, maybe what EBV does is it waits until the infected cell gets to the germinal center. Then this initiator transferase called 3B will become expressed, start putting marks on the viral genome, and later they'll be maintained by this system here. And this just shows you by an immunohistochemical approach, this germinal center in a lymph node that would have nice staining for DNA methyltransferase 1, but no staining for 3A. Now that's really excluded from the germinal center. It's really the 3B that's present there. So we looked by the methylation DNA immunoprecipitation approach, this time moving to a cell with latency 3 called GM12878. I think Majid knows quite well. It's a cell that's used by the ENCODE project very heavily. It's one of their tier 1 cell lines, but it's actually an EBV transformed lymphoblastoid cell line with latency 3. And so Remember, this is a cell that will have very low expression of DNA methyltransferase 3b. So we asked what happens if we express a control GFP gene, and looked at either the W promoter or the C promoters that control the EBNAs. And so if we overexpress 3A, it doesn't change the methylation too much at these sites. But if we now express 3B, which is normally silenced in the LCL, it really nicely methylates both the W and the C promoter. And what you see happen next is that it turns off the expression of EBNA3C. So if you follow on this Western blot, normally the LCL is making EBNA3C, one of the latency 3 proteins. It's getting shut off. As you can see on the Western blot, when we overexpress DNA methyltransferase 3b in the cells, EBNA2 goes off, LMP1 also is switched off. And so it sort of supports a model in which if DNA methyltransferase 3b switches on in a B cell as it moves into a germinal center, that could be a mechanism by which the virus starts to turn off the latency 3 program and move towards that more restricted state that's present in memory cells and Burkitt lymphoma cells. I know we're getting a little bit close to the end of the time. Just say quickly that we also bumped into the polycomb proteins as a maybe second layer of epigenetic control over the latent membrane protein promoter that might help us to understand how the cell could then move from latency 3 to 2 and from 2 to 1. We think that DNA methylation starts the silencing, and that additionally at the latent membrane promoter, there's an additional polycomb event that we think also leads to the final silencing of latency 2 to move it towards the latency 1 state. So in the interest of time, I'll kind of go to this model where putting together what we found in the CRISPR screen with what's known about the B cell development as these germinal center reactions take place. We think that EBV infects the naive B cell, initially starts to express this latency 2B program in which UHRF1 and DNMT1 are upregulated. You also upregulated an enzyme I didn't have time to tell you about called TET2 that actually takes methylation marks off DNA. So that would be kind of pushing the cells towards the latency 3 state where they're going to be in a lower methylation environment. But as these infected cells move into the germinal centers, now you'll start to have expression of DNA methyltransferase 3b naturally happen. I don't think it's known why that happens, but something about the microenvironment turns it on. So that can now initiate methylation marks on the viral genome. When it does that and starts to turn off EBNA2, TET2 would go off. So you now lose the enzyme that's taking off methylation at the right time, that you're starting to gain the expression of one that initiates methyl marks. And so then we think that as it then moves through the germinal center towards memory cell, there's also a step for polycomb to come in and turn off the LMPs and move the EBV genome towards this very silent state that for some reason occurs in Burkitt lymphomas as well as in the memory B cells. And so therapeutically, this could be interesting because there's a lot of interest in using these off-the-shelf T cells now, where you can, there are biotech companies that are storing huge banks of T cells that are matched for the right HLA, or so the doctors tend to call it HLA, and I think in immunology we call it MHC, or major histocompatibility complex. Each of us has a different system that presents peptides to T cells on our cell surfaces, and so you have to match the HLA type between what you have naturally and what T cells you would give somebody. And so there are these big banks that just have the same basically HLA alleles that we have. And so you can pick one of those off the shelf that has been raised to actually react with the latency 3 epitopes from usually EBNA3C proteins. And so basically, if you have a transplant patient who can't make their own T cells, or an AIDS patient, maybe you can infuse these T cells that are primed to recognize latency 3 expressing B cells and get rid of those cells. And so we're wondering whether that might be a future approach that could be used for Burkitt lymphoma, where currently you can treat Burkitt lymphoma in many cases, but with a very intense dose of chemotherapy. So much so that it's actually very hard to use in certain research limited settings, like in areas in sub-Saharan Africa, where it'd be very difficult to take people through this very intense chemotherapy that leads to immunosuppression and infection. And so there's room for a sort of a more gentle approach than this huge dose of chemotherapy that we currently give to Burkitt patients. And so in collaboration with Lisa Galino-Roth and Ethel Caesarman and Rich O'Reilly in New York, we were able to take a mouse xenograft model sort of through what we were seeing in cell culture. And so what we did was to add Burkitt xenografts. So you can basically grow the Burkitt tumor in immunosuppressed mouse so that the mouse doesn't just reject the tumor. And so you can grow these Burkitt tumors. And then instead of knocking out UHRF1, you can add a drug called decitabine, or here DCB, which is a hypomethylating agent. Basically does the same thing as the UHRF1 knockout does. It blocks the copying of DNA methylation at the level of UHRF1 and DNMT1. And so we could implant the tumors into mice, then infuse the decitabine drug to try to wipe out the methylation marks. And we could see that in some of the tumor cells, you could get re-expression of EBNA2. So you can see in these ones at the bottom, they don't express EBNA2, and so there's no brown color. But when we add the decitabine, you start to get the brown color at day 7 or even way out at day 19 in some of the tumor cells. And then if you follow the CD8 T cells that were then infused in the mice that were specific for the EBNA3 antigen and latency 3, normally they don't see the Burkitt tumor because it's not expressing latency 3. So in this row here, the T cells were infused without the decitabine, and so you don't see brown here because they're not homing to the tumor. But when you add the decitabine to take away DNA methylation, and you add the T cells, now you get a lot of brown color, meaning that the T cells are now homing to the tumor. And that was especially evident at earlier time points, but even out towards several months afterwards. And we could start to see some therapeutic effect where there was a luciferase gene in the tumor cell to give a readout for how much tumor is there in the mouse. And so in this gray line, you can see that tumor volume is going down a bit after decitabine, as well as T cell therapy has been given. Whereas if you just give decitabine alone, you can kind of stabilize the tumor, but not have as much of a loss of tumor volume as if you treat with decitabine and give the T cells. So it's sort of a beginning place to work on. Can we reinduce latency 3 in these tumors where it's quite silenced, and then open up the door to immunotherapy approaches? And so in summary, the screen really showed us that UHRF1 as well as DNA methyltransferase 1 were important host factors to maintain latency 1. This might fit with the biology in which UHRF1, DNA methyltransferase 1, as well as 3 are upregulated in germinal centers, and that all the domains of UHRF1 are important for this. UHRF1 binds to EBV genomes and specifically regulates the methylation state at key viral promoters, and that inhibition of either of these may increase the antigenicity of tumors by re-expressing the latency 3 antigens. And so I'll just skip some of the stuff I was hoping to have time for on metabolism crosstalk, and just go to the acknowledgements to say, I want to really acknowledge in particular Ray Guo, who's a very talented postdoc who ran the CRISPR screen I told you about today. A lot of help from Yuchen Zhang in the lab, a graduate student who recently graduated. We often work closely with Bo Zhao across the hall, and at Weill Cornell Medical Center, we did the xenograph studies with Lisa Athol-Caesarman and Rishi Puri in the animal core. And at the Broad Institute, John Dench is a wonderful resource for anything CRISPR. He's the one who develops these libraries, and is a great resource. So he's happy to talk with anyone. You can just email him, and I think if you have questions about screenings, he would engage and often collaborates with people from afar on CRISPR screen projects. And we work closely with Vamseen with Lab at Mass General on metabolism projects. So at this point, I'd like to stop and just take any questions in the few minutes remaining that you guys might have.
Thank you so much, Ben. That was amazing. After over 50 years, we still learned a lot of new stuff from the EBV field.
Sort of an old virus given new life.
That's wonderful. So any question from audience, please feel free to unmute yourself.
Yes, I have a question. So nice presentation, Ben.
Thanks.
I really like the historical intro.
Yeah, it's sort of fun.
Yeah. So just this is a very general question. What's the treatment landscape for EBV right now? And are there inhibitors for UTHR?
Right. No, those are great questions. So the main problem for EBV treatments is that we have a good drug called acyclovir, or a cousin called ganciclovir, that acts on the lytic cycle. So another interest that we have is reawakening the lytic cycle to sensitize the tumor cells to those drugs, because in that case you could wake up the lytic cycle and then come in with a nice drug, very specific for the cells that have lytic EBV. But in general, all the EBV-associated cancers have latent EBV, so you can't attack it with those drugs. And so that's where we're stuck with. Usually people are treated with the same chemotherapy that they would get if it was a tumor that didn't have EBV, so whereas in sub-Saharan Africa, you'll almost always have EBV driving the Burkitt. In the West, only about 30% of the time you find EBV in the tumor. And so we treat everybody the same with this huge chemotherapy. For some tumors that are B-cell tumors, there's an antibody called rituximab that just wipes out the B-cell compartment almost. It's recognizing an antigen on the B-cell surface and just takes out good and bad B-cells together. So that's a typical thing that will be done for EBV B-cell lymphomas, that you may wipe out B-cells generally with an antibody together with chemotherapy. Really have limited therapies on the epithelial side. That usually the case is if you find them early, they're sensitive to chemotherapy and radiation, but if they're detected later, they become very resistant. And so there's not good therapies for the more resistant form of tumors that can evolve in the areas of nasopharyngeal cancer and gastric cancer. So that's part of why we're interested in how we could change the epigenetic state of the tumor genome so that we could harness the presence of a viral genome in the tumor cells. Each tumor cell has many copies of the viral genome, and so if we could change it, then maybe that would allow us to more specifically go after the tumors than what we're doing currently. In the case of mono, they actually don't have a specific treatment because it's so much driven by latent EBV that the antivirals don't work very well. And so you just sort of wait for the immune response to kick in.
Great.
I have a question.
Yeah.
Hi, Dr. Ben. This
Hi.
is Rishni. Great
Hi.
talk.
Yeah,
Great
thank you.
talk. So I had a question regarding the decitabine treatment of the xenografts for Burkitt lymphoma. So for the CTLs that you guys used, was it specific to a certain antigen? Because BL would be just EBNA1, right?
Yeah, no, exactly. So that's where this, so Richard O'Reilly, actually his lab at Memorial Sloan Kettering developed these huge banks of T cells that are now sort of being commercialized. So we had a T cell that matched the right HLA type in that Burkitt tumor that we're using in the xenograft, and then it also looked for a peptide from one of the EBNA3 proteins. And so I think that's why it's not homing to the tumor until we add the decitabine. It can't find the EBNA3 protein, but then when you add the decitabine and the cell starts to make some EBNA3, some of it gets chewed up and presented on the MHC on the cell surface, and then
makes sense.
So yeah, that's sort of the idea. But that's also why you need these off-the-shelf approaches. It can take months. If you were to harvest the person's own T cells, you could develop in cell culture these activated T cells just by growing them on the lymphoblastoid cell line, but it takes months to do that.
Yeah, the reason I'm asking is because sometimes when we have like tumors, which are like cultured long time back, like stomach adenocarcinoma tumors, then if you want to find out a CTL specific for those tumors, would they be able to do using a similar approach? Would that be possible?
Yeah, I think yes. So yeah, I think so. I think it potentially could be. And then there's neat approaches now to identify what is the T cell receptor. And in some cases, they can tell if the T cell receptor is actually anti-EBV. That they've gotten good enough at sequencing by next generation sequencing and recognizing certain receptors that can form if they're anti-EBV.
Got it. Thank you so much.
Yeah, so it's interesting.
Wonderful. Any other questions?
Hi, this is Cheetah Das from chemistry.
Hi.
I have a quick question.
Yeah, sure.
So it looks like the URH, the virus is hijacking the host E3 ligase. So is the ubiquitous and reversible? Because I know EBV also.
It's good. Yeah, it reminds me, I forgot to answer part of the other question because there was a question about are there inhibitors? There are like inhibitors in development as sort of tool compounds at this point. I think it is reversible. I'm trying to think though, there must be a deubiquitinating enzyme that would come in and take that off. I'd have to look up if that's known, what the deubiquitinating enzyme is that reverses the mark.
Yeah,
but I think it is reversible.
I think also expresses a DUB. But it may be involved in different pathways, but I don't know.
Yeah, you're right. In the lytic, right, true. Yeah, so we haven't looked at whether that could be at all. It's interesting. I didn't really highlight it here, but it looks like the cells kind of have a choice that if you're in latency 1 and you hypomethylate, some go to the lytic cycle and some go to the latent 3 state. It's almost like the latency 1 is this very repressed state, and from there it has a choice to spring to 2 other places. It can go lytic or it can go to latency 3. if you take off the suppression, it moves to one or the other. So there are some lytic cycloantigens made in some of the cells that could maybe turn on the dub that the EBV encodes.
Okay.
Yeah.
Right. Thanks.
But yeah, so I think there's interest in developing UHRF1 inhibitors, but they haven't really been taken all the way to, I think, clinical testing at this point.
Wonderful. Are there other questions? So let me ask one. What about like Ben, the DNMT inhibitors? Have you tried any of them? Do they make like these Burkitt lymphomas more immunogenic?
Yeah, I think they do. Yeah, that's what I was trying to show you with that xenograft. I think they do. You know, the neat thing in that scenario is we can add the decitabine, or there's another one called 5-azacitidine you can add, and then come in afterwards with the T cells so that they don't themselves get inhibited by the the hypomethylating agent. So there was because otherwise you can sometimes have the challenge that there's a double-edged sword that the agent that changes the EBV latency also can suppress the T cells.
And do you think that ICAM1 kind of bring in and activate the T cells? Is that?
Yeah, no, I think that's part of why this might work well. That in addition to reactivating the EBNA3 expression, you really make the B cell into a very nice T cell activator. So the latency 3 program upregulates ICAM1 that can help sticking to other cells and trafficking to the right place to find T cells. You get more of the HLA expressed in the cell surface and more of the B7 molecules that activate T cells. I think it's why CAR T cells have been so successful against B cell cancer so far, and really not others, is that the B cell is a great activator of T cells. And so if you're using this chimeric antigen receptor T cell approach, it's going to work really well against B cells, where you've got a B cell that's able to really talk to that T cell and activate it. And so I think that's true with the latency 3 program also. EBV has evolved a way that it doesn't want that cell to turn into a lymphoma cell and kill the host. It wants to escape and become a memory cell, and then infect you for your life so that you can eventually spread it to somebody else. And so I think it's made that latency 3 program very visible to T cells. So it's very briefly made and not going to cause a cancer most of the time.
Wonderful.
Yeah, so I think so.
Thank you so much. Any other question?
I do have one last quick question.
Yeah,
I
sure.
won't take up too much time.
No.
So, the ICAM that you have seen in the B cells, do you assume or do you know if that's similarly seen in epithelial cancer cell lines as well?
Yeah, that tends not to be. Yeah, I wish it was, because we would use it for other screens if it was. I think it's more of a, at least, I don't know if it's B cell specific, but more hematopoietic marker.
Okay.
I think normally it's used by cells for trafficking and how they'll stick at certain phases in the lymph node in certain places. So I think that's, I know neutrophils will make that as part of what regulates some of their interactions with the vasculature. But so I think there's probably other things that might change in the epithelial side.
Thank you.
Yeah, no, that's a good question.
All right, so thank you so much, Ben.
Thanks for hosting. Thanks for the invitation today, Majid.
Let's thank our speaker once again, and hopefully next time we'll see you in person.
Yeah, right.
Thank you. Thanks so much.
Okay.
Bye.
Okay, bye.
Thanks.